Good morning, everybody, and welcome to this session of Plants, Pests, and Pathogens. Just the same reminders as usual. This is a slightly updated version of Collaborate, but I think all the navigation will be the same as what you're used to. Lee J will lead you through some of that in a minute. But before we go there are a couple other things I wanted to mention, and that is for those of you who signed up for LMS credit last time, make sure that that credit has been recorded. I actually didn't do that until last week, so be sure and check on that. And likewise, for this session, be sure and check on that. It'll take me a while to do that. Basically, I have to wait until the recording is posted and then just go through the recording and look for you. And so while we're on the subject, make sure that your sign-in name is a name that I can recognize as being you. And as always, we ask that you record the number of members attending in your county. All right, so you're listening to me. I'm Barbara Shue, Extension Plant Pathologist and Director of the Plant Disease Insect Clinic, sitting in for Lucy Bradley today while she is enjoying her sabbatical in Costa Rica. I think we're ready for Lee J to take over with some information on how to run your Collaborate session.
Good morning, everyone. My name is Lee J. Temple, and if you have any technical questions, you can direct them towards me. The version of Collaborate has been upgraded, but the navigation tools are pretty much the same. At the top, you have your audio and video window. There is a talk button. If you have a question that you would like to voice, you can click the talk button and we will be able to hear you. Under that, there is a window for participants. You have different emoticons that you can click to share. So if you're wanting to laugh or applaud, or if you have a question, then you can click on those. If you have a question and you would like to speak with us, click on the hand icon and it will alert us that you have a question. And that's where it is right there. At some point during the session, you will be asked yes or no questions, or maybe to choose from A, B, or C. And you will do that by clicking on the checkbox right below your name. If you see that, go ahead and click the green X to let me know that you see it. Okay. And below the participant window, there is a chat box. If you don't have a microphone or you just want to text in your questions, type in the chat box. It is supervised, so we can see anything that you type. So if you talk with one of your buddies over the chat, we'll see it all. If you have any questions, type them in the chat box, raise your hand, and we'll get to you when we can. Thank you. Okay, so if everyone will mark on the map where you're listening to the session. I'm in Wake County, so I'm going to click on the starburst so I can click on the Wake County part of the map. All right, let's get started.
All right, our featured presentation this morning will be from Liz Meyer, talking about plant propagation. Then later we'll hear from Matt Bertone on insect issues and Mike Munster on plant disease issues. Again, we are changing over from oh gosh, I forgot what it was called. we're changing over the featured plant feature. A new featured plant feature will start with the next Collaborate session. So ready to hear from Liz. She is a lecturer in the Department of Horticulture Science. She earned her master's degree here at NC State, focusing on ornamental plant breeding and propagation. And she is now an instructor in the department. She teaches World of Horticulture, home plant propagation, home landscape maintenance, and plant propagation. So Liz, we're ready to hear from you. I'm sure that everybody is really excited to hear about plant propagation.
Good morning, everybody. I'm glad you're excited to hear about plant propagation because I'm excited to talk about it. I see a couple of familiar names on the list here. Hi, Amy. Amy was my teaching assistant for my home plant propagation class last spring. And there I am. So this morning, I just want to talk to you guys about just sort of a general overview about plant propagation. I actually teach 2 classes on plant propagation here at NC State, so I can talk for an entire semester on plant propagation, but for you guys, I'll try to keep it to around 30-40 minutes so you have some time to ask questions. Usually when we talk about plant propagation here in the horticulture department, we talk about how it's both an art and a science, because to really be a successful propagator, you have to master the technical arts involved, technical skills like grafting or even taking a good cutting. But it's also important to understand all of the science behind it, so you know why you're doing what you're doing. And so, again, this is just going to be a pretty general overview of those 2 things, but hopefully helpful information. So a few basic requirements for success. The first being choice of plant material. It may seem pretty common sense, but when you're choosing to propagate a plant, you want to pick a nice, healthy plant. You don't want to choose a plant that has obvious disease or insect problems, because you're just going to transfer those to your new plant, and it's going to be hard for that plant to even successfully propagate in those conditions. You want it to be well watered, have good nutrition, be receiving adequate light, all of those things. Basically just a healthy plant is what you're looking for. For the growing conditions, it doesn't matter which type of propagation you're doing, what technique you choose. These 3 things are always important for growing conditions. Sanitation, excluding pathogens and pests as much as possible, correct moisture levels, and correct temperature levels. And I'll talk a little more in detail about those as we go along. And then finally, you need to know general knowledge about propagation techniques and specific knowledge about plants. Every plant species has a certain time of year, a certain propagation technique that is best suited to that plant species. And that's something where having a really good resource is going to help you. And I'll give you guys some examples of those as we go along. So I just want to talk about sanitation a little bit more because it is so important. When you think about propagation, you should think of it essentially as you're caring for this little baby plant that doesn't really have much defenses. So excluding pathogens and pests from the propagation environment is so important because that plant is tiny and can't really fight it off much on its own anyways. So you want to start with a media or substrate that is free from pests. If you're buying a sealed bag of media from the store, it's already been pasteurized, so you don't really have to worry about this. But it is something to bear thinking about. You don't want to try to take soil from outside to use as your media for propagation for a variety of reasons, but mainly because there's a lot of pathogens already present there. As I mentioned before, you want to make sure your stock plant, the plant that you're propagating for or from, doesn't have any disease problems. You want to disinfect basically everything you're using. Your containers, if you're working in a greenhouse or on benches, those need to be disinfected. Any media bins, any tools. And usually we do this with each rotation of propagation. So here at NC State in the greenhouse, we have a propagation class out there each semester, so we sanitize everything in between semesters. And a really easy thing to use for sanitation of these things is 10% bleach. There's a lot of different ways to sanitize things, but 10% bleach, you can't really go wrong with that. And then finally, general sanitation. This has to do with things like, let's say you're taking cuttings. If you drop a cutting on the floor, you don't want to pick that back up and stick it in your flat or wherever you're propagating, because it's just picked up what other spores, bacteria, etc. could be on the floor. So just things like that. All right, there are two major types of propagation, sexual and asexual. You probably often hear asexual propagation referred to as vegetative propagation. They're the same thing. For sexual propagation, there's only one method, and that's seed, of course. For asexual, we have several methods: cuttings, layering, grafting, and tissue culture. I'm not going to talk about tissue culture this morning just for the sake of time, but we will talk about all of the other ones. Now, if there's thinking about the advantages of each of these when you're getting ready to propagate a plant so that you can decide which one best suits your needs. Sexual propagation or seed, the benefit of that is you don't really need any technology, and by that I mean you don't really need any special equipment. You don't need any kind of structure to house your seeds. You don't need mist. Of course, those things can help, but basically you can go stick a seed in the ground and it'll grow. I mean, that's simplified, but possible. So you don't need a lot of technology. They're very easy to ship if we're talking about the commercial aspect of things. Seeds are easier to ship. And when you sexually propagate a plant, you're getting genetic diversity. So sexual propagation is what plant breeders usually use, because by doing this, we're getting offspring that are different from the parent plant. If we're a plant breeder, different is what we're looking for. Asexual propagation, on the other hand, you'll see you get genetically identical offspring. So if you don't want different, if you want offspring plants that are exactly the same as the parent plant, then asexual propagation is the way to go. And this is what's used commercially a lot because, of course, we like to have uniform plants for sale in the nursery industry and things like that. Other benefits of asexual propagation: we get a bigger plant faster than we do from seed. You can propagate plants that don't produce viable seed because, especially with breeding for plants that are non-invasive, we're getting more and more plants that actually don't produce fertile seed. So in that case, we have to propagate them asexually. That's really the only option we have. And then finally, we can avoid juvenility. This has to do with woody plants. Woody plants all go through a juvenile stage where basically they're not going to produce any flowers or fruits. They're just producing a lot more stems and leaves and roots. For something like a fruit crop, apples for example, the juvenile period can be quite long. If you plant an apple from seed, that could be 7 to 8 years before you actually get apples on your tree. So by using asexual propagation, if we propagate from mature plant tissue, we can now just skip that whole juvenile phase and get a plant that produces fruit in maybe 1 to 2 years. So with that, I'm going to jump into specific types of propagation, but I just wanted to mention if anybody has any questions or anything while I'm talking, please feel free to jump in. One thing real quick before I do, this slide is just basically I'd like to explain that sexual and asexual propagation are usually used in conjunction in the nursery industry. So if you look at the flowers in this picture, these are all, of course, hellebores. On the top left, we have a flower that, like a typical hellebore flower, it hangs down. And it has really nice coloration, but it's hard to see that when it's out in the landscape because the flower is pointing down towards the ground. On the right, we have another hellebore. It doesn't have quite as nice of a coloration, but the flower is more turned up so we can see it better. So wouldn't it be great if we could combine all those traits into one flower? And so this is where we'd use sexual propagation. These are actually hellebores that were bred at Pine Knot Farms, which grows hellebores. So these top two flowers are the parents. They were sexually propagated, and what we got is the flower in the bottom there. You can see it has the nice coloration of the flower on the top left, but it also points upwards so we can see it better like the flower on the right. And so we've got the best of both worlds. So we use sexual propagation to get that flower, but now that we have what we want, this is when you'd use asexual propagation to make a lot more of that exact same thing. So again, commercially, the two methods are usually used in conjunction with each other. And there it is. All right, so let's start with sexual propagation, or seed. The first thing that you need to make sure of when you are propagating by seed is that your seed is viable. That means it's alive and it's capable of germinating. All right, so you want to make sure it's gotten fully ripe. A good way to test for viable seed actually is to throw all of your seed in a jar of water, and usually the ones that float up to the top are not viable. They float to the top because there's nothing in them. There's no embryo in there, and so you can just skim those off and plant the seeds that sink to the bottom, and you can be pretty solid that those are viable seed. You do have to have appropriate environmental conditions. Needs to be moist. The media needs to be moist but not soaking wet. You need to have fairly warm temperatures, usually between 50 to 70 degrees Fahrenheit. You have to have adequate oxygen circulation. This kind of goes back to the water. If you have your soil completely saturated all the time, there's not going to be adequate oxygen in your seed germination environment. And sometimes you need light. This depends on the species. Some plant species, the seeds require light to germinate. Some plant species, the seeds actually require darkness to germinate. So that's one of those species-specific things that you really need a good resource to tell you about. So what happens basically for all seeds once you plant them for them to germinate is a three-stage process of germination. In that first stage, we have what's called imbibition of water, which just means the seed takes in water. That's the very first thing that has to happen. So no water, no germination. Stage 2, if you're just watching the seed, you're not really going to see anything happening in stage 2. Everything is happening inside that seed. But basically, enzymes are activated within the seed. They start digesting energy sources and sugars inside that seed and move that energy to the growing points so that what we think of as actual germination, where things emerge through the seed, begin to happen. And so that's stage 2. Again, if you were looking at the seed, you wouldn't actually see anything occurring. And then that brings us to stage 3. Cell division starts to happen at those growing points, and we get things actually emerging from the seed. And we have, of course, because plant scientists love words, we have special names for all of these things. So the first thing that typically comes out is what we call the radicle. It's just the seed root. And then after that, we get the hypocotyl, which is the seed shoot, followed by the cotyledons, or the seed leaves, leaves that have already been formed inside that seed. And then finally, the epicotyl, which is just more seed shoot that has what we call the first true leaves on it. So basically, stage 3 is when things actually start to come out of the seed. So that's our general sequence of events for germination. How deep should you plant your seed? You can base this on the size of the seed. The general rule that we use for that is the seed should be planted 1.5 times as deep as the diameter of the seed. So the larger the seed, the deeper you're going to plant it. It's really important not to plant your seeds too deep because if you do that, take a very tiny seed like that lettuce seed. If you plant it too deeply, it doesn't have enough stored energy inside that seed to germinate and make it up through the surface of the media if you plant it too deeply. Spacing, you don't want to space your seeds too close together when you sow them. This is, if you have a seed packet, it's going to tell you on the seed packet actually how close together to put those seeds. So the picture on the left here, you can see what happens when you sow your seeds too closely together. They have to really compete for light because they overlap each other too much, and so they become very tall and leggy. They're weak seedlings, they're going to fall over, and there's really no going back from that. Once that's happened, you can't really fix it. So spacing is really important. The picture on the right here is a pretty good example of spacing. There's not a whole lot of overlap of those leaves. All right, so let's say all of that has gone well, your seeds have germinated. How do we know when to transplant them? Again, the general rule here is when the first set of true leaves emerge, that's when you transplant. So again, when the seed first germinates, you're going to get those seed leaves coming out. They don't really look shaped like the actual adult leaves of the plant. They usually look just kind of round. So for example, in this picture here, this is a seed leaf. Let's see, let me use the little star. This is a cotyledon, this is a cotyledon. All right, and those first true leaves are here, and here, and here. All right, so once that first set of true leaves emerges, then you can start thinking about transplanting your seedlings into their next location if you didn't directly sow them there. The most important thing to remember during this stage is to be very gentle with the seedlings. You never want to pull a seedling out from the top. You always want to use some sort of tool such as those pictured here to scoop up under the seeds so that you don't damage the roots. This is a really crucial step in seed propagation that is very easy to mess up. So you want to take your time and very gently scoop the seedlings up from under the root system and then very carefully plant them into their new container. Now, in some situations, you'll have done all of these things right and the seeds don't germinate. And the reason that occurs is because there's some sort of seed dormancy. Now, we don't have time for me to go in a lot of detail about this, but basically there are two major types of seed dormancy. Seed coat dormancy, in which basically your seed isn't germinating because it has a really hard, thick seed coat. Usually what happens in this situation is that first step of germination can't happen. There's no imbibition, there's no water getting in the seed, so germination doesn't occur. And then we have embryo dormancy, which is within the seed. Basically, both of these major types of dormancy, and I have subcategories listed below, which we don't really have time to go into, but basically what this means is these seeds will not germinate unless they've been exposed to a certain type of treatment. And the whole reason this exists in nature is to keep seeds from germinating at a time of year when the environmental conditions might be favorable to germination, but it's not a good time. So for example, a lot of your trees and shrubs are going to set seed in the fall, and especially here in North Carolina, it could be plenty warm enough and wet enough in the fall to support seed germination. But we don't want those seeds to germinate in the fall because winter is coming. That will be too harsh for the seedlings and they'll die. So a lot of times we have seed dormancy in those types of seeds to keep them from germinating right away. If we have a seed coat dormancy, all right, so something about the outside of the seed is keeping it from germinating, we have to do a treatment called scarification, where basically what you're doing is just making a hole in that seed coat so that water can get in. You can do this by filing it with a file, cutting it with a knife, rubbing it with sandpaper, or sticking it in very hot water. You don't want to stick it in boiling water because then you're going to cook the seed and it's going to die, but just below boiling temperatures. Or you can soak it in sulfuric acid. This is not something, of course, you want to do outside of a lab. So if you have a seed with a very hard seed coat and it's not germinating under normal conditions, you might try a scarification technique. You may also remove the whole seed coat, but you don't want to have to do that unless it's absolutely necessary. If it's embryo dormancy, a dormancy inside the seed, all right, so it's not the seed coat keeping it from germinating, but something inside that seed, you want to expose it to a stratification treatment. And this basically just means you keep those seeds at a certain temperature and a certain moisture level. Warm moist or cool moist stratification, most of the time, especially for a temperate area like here, you're going to be using cool moist stratification. And essentially what you're doing in this scenario is simulating winter. All right, so all you do is you take your seeds, put them in moist media, again, just moist media, not sopping wet or they're going to rot, and then store them somewhere between 33 to 50 degrees Fahrenheit, usually for a period of 3 to 4 months. So you can do this in your refrigerator. You can also do this by burying the seeds outside and simply letting them experience winter. But either way, that would be considered stratification. As far as what kind of dormancy your seed has, or if it has it at all, this again is a very species-specific situation where you'd want to look it up in a resource to find out. All right, so now we're going to move on from sexual propagation to asexual, then discuss cuttings. I imagine, like seeds, most of you are pretty familiar with cuttings, so we're going to sort of breeze through this. The most familiar type being, of course, stem cuttings, such as these Leyland cypress here. With the stem cutting, we have 4 major types. An herbaceous stem cutting is just a piece of stem cut off of an herbaceous, non-woody plant. And with all of these stem cuttings, we're going to be using a section of stem that has at least 2 nodes, 2 areas where either buds or leaves are coming out. At least 2 nodes on that portion of stem. So herbaceous stem cuttings are, just as the name would suggest, herbaceous-only plants. And then for woody plants, we have the categories of softwood, semi-hardwood, and hardwood. And those 3 categories are referring to the firmness of the wood and also the time of year. Typically, softwood cuttings are going to be found on your plant in late spring and early summer, early to midsummer. Their stems will still be very pliable. Semi-hardwood is going to be found in the fall mostly, or late summer. This is when your stems start to firm up a bit, and then if you're taking it into winter when the stems are dormant, that would be considered a hardwood cutting. And that's what you see in the picture here. For most cuttings, you need to keep them in a very moist environment, usually a humidity tent or an intermittent mist. Hardwood cuttings, because they're dormant, you can actually stick them just outside as they are in this picture, which makes it a little easier. So the basics of how to take a stem cutting, you want to make sure that your knife or pruners are very sharp. If they're not, when you make that cut, you're going to crush some of the tissues of the plant and it's not going to root as easily. So again, you want to have at least 2 nodes and at least 3 inches long piece of stem. Cut it at an angle. Remove any flowers or fruits. There's 2 reasons you do this. One, usually flowers and fruits are going to fall off your cutting and rot in the media, which isn't very good. And also in some cases, they can, if it's a flower that's just starting to bloom, they sort of drain energy out of your cutting, which you want all that energy for roots. You're going to strip the lowest leaves from the cutting base because, of course, we're going to stick the base of that cutting into some media. We don't want those leaves to rot. You're going to cut the leaves in half. This is particularly important if you don't have an intermittent mist system because the larger the leaves, the more water that cutting is losing. It's just transpiring out of the leaf, and it doesn't have any roots on it, so it can't take up more water. With cuttings, your whole thing is you're in a race against time. You want roots to grow on that cutting before it completely loses all its water through the leaves. So by cutting those leaves in half, we reduce the transpiration area. Now you may be thinking, well, why don't we just pull all the leaves off? You do want to retain some part of the leaves just so that they can continue photosynthesizing and producing energy for that cutting. In addition, reducing the leaf size for some really large leaf species is going to help you fit more cuttings in the space that you have for propagating. So it's an important step. All right. Next, you'll apply rooting hormone. Most cuttings will benefit from using rooting hormone. Now, rooting hormones are basically in a class of hormones known as auxins. So when you go to buy a rooting hormone from the store, make sure that you see something like IAA, IBA, or NAA in the ingredients list. The active ingredients should have something like IBA on it, which will probably be spelled out as indole butyric acid. If it doesn't have that, if all you see when you look at the ingredients is your general NPK, then it's not a rooting hormone. I don't care what they say on the bottle, it's just a fertilizer. All right, so make sure it has one of those auxin sources in it. You can buy rooting hormones in two different forms. One is a talc powder form, just like the one shown in the picture here. There's a lot of different brands, Take Root, Rutone, Hormidin. They're all the same thing. They just have different percentages of auxin in them. All right, so talc powder, it's readily available. It's fairly inexpensive. It works really well on herbaceous stem cuttings and easy to root woody species. However, for your more difficult to root woody species, you're going to want to use a liquid hormone. Again, it's going to have the same active ingredients such as IBA, but it's in a liquid form and usually at a higher concentration. Again, there's a lot of brand names for this listed here, Dip and Grow, Seaman K, etc. Mostly the same, just different concentrations of auxin. So again, that liquid works better on more of your difficult to root woody species. In addition, for some really hard to root species, you can do a little extra wounding at the base of the cutting, basically just taking a razor blade and scraping off a little of the bark on either side of the base of the cutting. This can help encourage rooting for hard to root stem cutting species. And then finally, of course, you're going to stick your cutting into the media. It's best if you can have a node in contact with or just below the media surface, if at all possible, especially if it's a hard to root cutting species. So those are our basics of how to take a stem cutting. And then you need to put it somewhere, again, where it's going to be in high humidity situation. Commercially, this is done in intermittent mist systems as shown on the left here. But you can do it in simpler ways. On the right, this is actually some cuttings that a student of mine took for home plant propagation. So they just have a pot with the cuttings in it and plastic bag over the top to help hold in the humidity. So you don't necessarily have to have a greenhouse or a mist system. They, of course, help, and you're going to have higher success with that. But if you can somehow keep those cuttings in as close to 100% humidity as you can, you'll be fairly successful. Stem cuttings are the most commonly used commercially for propagating plants asexually. There are some plants that can be propagated by leaf cuttings and root cuttings. So for leaf cuttings, they could be leaf blade only, such as the picture on the right here of the snake plant. So you're just cutting off a piece of the leaf blade, treating the bottom with rooting hormone and sticking it in media, and you can see the roots that are forming there. In addition, this is a new shoot that's starting to form. Leaf blade and petiole would be like the picture on the left here. This is an African violet leaf. So we have the leaf blade and the leaf petiole, and you can see the new plants coming here at the base of the petiole. Leaf blade and petiole leaf cuttings are generally only going to be able to be used on herbaceous plants, and often it's used for a lot of house plants, as you can see here. Woody plants will never work from a leaf blade only or a leaf blade and petiole cutting. For a tree or shrub, you're going to have to use what's called a leaf bud cutting if you're not doing a stem cutting. Here's another picture just of a leaf blade only cutting. This is a Rex begonia leaf. It's placed flat on the surface of the media, and then the new plants come up through that leaf. All right, those leaf bud cuttings, again, the only type of leaf cutting that will work on a woody plant. Here you're going to have the leaf blade, the leaf petiole, and a small section of the stem. And that section needs to contain a bud here at the base of the leaf. All right. Now you may think, why not just do a stem cutting? Well, because they're so small, you can get a lot more leaf bud cuttings from a plant than stem cuttings because you only need essentially one node. So if you're trying to propagate a plant where you don't have that much stock plant material, a leaf bud cutting is a good option. All right, and this is a picture of a camellia. Camellias are a good example of a woody plant you can propagate by leaf bud cuttings, and you can see here the original leaf, the piece of stem that had the bud in it, and here's our new shoot coming out from that bud and the new roots. Just like there are a limited number of plants that can be propagated by leaf cuttings, there are also a limited number of plants that can be propagated by root cuttings. So for a root cutting, again, you just be taking a small piece of the root, sticking it in the media. You actually don't need rooting hormone on a root cutting. And it will produce shoots first, as shown in this picture. Eventually it will produce roots and a new plantlet. But again, a much smaller group of plants that we can propagate this way. All right, so given the amount of time we have left, I'm going to go through layering, and then I think we'll have to stop because I don't think we're going to have time to get through all of this, which I thought might happen. So layering is a type of asexual propagation that's not commonly used commercially just because it's very labor intensive, but it's a great technique for homeowners, especially for plants that are difficult to propagate by cuttings. With layering, basically what you're doing is you're getting that cutting to root while it's still attached to the parent plant. And the benefit of this is while those new roots are forming, that plant is still getting all the water and all the energy from the parent plant. When you take a cutting, it no longer has access to those things because it's separated. So there's several types of layering. One very common and easy way to layer plants is called air layering. There's basically two major steps. The first one in the picture on the left here, you simply girdle the stem, and by that I mean you scrape just the outer layer of bark off on a 1 to 2 inch section all the way around the stem. Then you just cover that area up with media, moist media, seal it up with plastic as shown in the picture here, and what will happen is roots will form in that soil. It usually takes about 3 months for this to happen, and then once those roots are formed, you cut the plant off below that root ball and you have a new plant. Pretty simple. Of course, even more simple would be simple layering. This works well with some of our shrubs with very low branches. You simply peg a section of the stem down in the soil, leave it for about 3 months. It'll form roots in the covered portion. And again, you can just cut it off below those roots and you have a new plant. Serpentine layering, very similar to simple layering, but in this case, this is something you can use for plants that are vines. Grapes, for example, work really well with serpentine layers. So you're just pegging that branch down and covering alternate sections of the stem. Roots will form and you have a lot of new plants. And you can even do it right in the pots you're going to grow them in, such as the picture here. Tip layering, very similar to simple layering, but this actually occurs naturally on a lot of your bramble fruits. So blackberries, raspberries, that sort of thing will already begin to arch their branches down towards the soil at the end of the season. And so you simply bury the tip of that branch and a new plant will form there, which you can then cut off. And then finally, mound layering. This is actually the only type of layering that is still used commercially. In this type of layering, what we do is we actually cut off the entire plant about 6 inches from the ground and then cover it up with media. After a couple weeks, new shoots will begin to grow up through that mound of media and we cover them again. And we keep doing that until a sizable mound has formed, and within that mound, as you can see in the picture on the right, roots are going to form on the stems, and then you can cut each of those branches off and you have a new rooted plant. All right, so I'm going to stop there. I apologize we didn't get to go through everything, and I know I really whipped through that. One thing I did want to get to.
I think we have time to go on just a little bit if you'd like.
Okay. We'll go through one more section. The asexual propagation by division and specialized structures, since we have the time. Again, division is something that I'm sure you're all already familiar with. It's not just a propagation technique, it's also a maintenance technique. Some plants just grow so densely that you need to divide them periodically for the health of the plant. But with division, you're essentially cutting the plant into sections, and the main thing about division is just that each section needs to have both roots and shoots on it. You're only limited with how many new plants you can get by division based on how big the plant is. All right. But very closely related to division, we have division of what we call specialized structures. And this applies to plants that have bulbs and tubers and things of that nature. These are some techniques that not everyone is familiar with, but that can really help you get a lot of plants from a single bulb. So I have two pictured here. The one on the top right, twin scaling. This is a technique in which basically you're going to take a bulb and cut it up into many pieces. The key is that every piece that you cut up has to have a part of the basal plate attached. And the basal plate, if you look at a bulb, is just that thickened area on the very base of that bulb. So as long as every piece has part of that thickened basal plate in it, you're fine. So you cut that bulb up, throw those things into a Ziploc bag full of something like sand or perlite with really good drainage. You want to have it barely moistened, and that's all, because it's really easy for this to rot. So you take those bulb pieces, put them in a bag with perlite or sand that's been barely moistened, and then put it in the dark for around 7 to 8 weeks. It works best if you can treat your bulb pieces, soak them in about 30 minutes of fungicide treatment first. But you put them in the dark for 7 to 8 weeks, and what you'll get is tiny bulblets like these shown in the picture. It's quite an old picture. But these are tiny bulblets, and so those will form and you just plant them out like a regular bulb. So from one bulb you can end up with anywhere from 20 to 50 bulblets forming. Somewhat similar is what's shown in the picture on the bottom left here, basal cuttage. In basal cuttage techniques, basically what you're doing is either cutting or completely removing that basal plate. Again, that thickened area at the bottom of your bulb. So in this picture, what they've done is completely removed it. Then again, they put that bulb in a bag with slightly moistened sand or perlite, left it for 7 to 8 weeks, and you can see how many bulblets have formed on the base of that bulb. And again, you just attach each one of those and plant them out like a regular bulb. This is particularly nice for more expensive bulb species, specialty bulbs that you may spend $20 on a single bulb. Really nice way to propagate those and make the best use of your money. Corms, very similar to bulbs. This would be plants like gladiolus, crocus. Those both have corms. Corms actually naturally produce tiny mini corms on the original corm. They're called cormels, and you can see a bunch of them here. Again, you just attach them and plant them. So this is something where you just keep an eye on your corm, maybe dig it up periodically and break off any of these cormels that have naturally formed. You don't have to do anything to make them happen. They just happen on their own. And then tubers and tuberous roots. Most of you are familiar with seed potatoes, which aren't actually seeds, but rather an asexually propagated potato. The key for propagating a potato or any other type of tuber is to make sure that you cut it up into pieces and that each piece has at least one eye or one bud on it. It has to have that bud on it for a new plant to form. But you cut it into pieces with at least one bud or one eye, plant those out, and they will each form a new plant. Very simple, very cost effective. A tuberous root, on the other hand, such as a sweet potato, is not going to have those eyes or roots on it. What you can do with a tuberous root, of course, you can just plant it, but a way to get even more plants off of a tuberous root is to lay it on a tray of media, as shown here. And what will happen is it will produce several shoots, which we call slips. Each one of those can just be cut off of the sweet potato and planted somewhat like a cutting, and it will root very quickly. So sweet potatoes and basically any other type of tuberous root, cassava for example, you can propagate this way. And then finally, there are certain plants that have other specialized structures that we can use for propagation. So on the left here, we have bamboo, and this is called a culm. It's just a specialized stem where you can cut off a section, lay it in some media, and it'll grow a new plant. Very much like a stem cutting, but unlike a stem cutting, you don't have to worry so much about it drying out because it doesn't have any leaves originally. This works with bamboo, which why you need to propagate bamboo, I'm not really sure, but it also works with a lot of our ornamental grasses that maybe you would want to propagate. And then finally here on the right we have something called a pseudobulb. This is a specialized structure only found in orchids. So if you look at a lot of orchids, they'll have this green swollen base. That's a pseudobulb. And you can actually in some cases break those off, one or two of them together, stick them in a bag of moistened media such as sphagnum moss, and they'll begin to grow a new orchid plant just from that pseudobulb. So specialized structures, very easy way to asexually propagate a plant, very cost effective. Of course, it has to be a species that produces that particular structure. All right, we definitely don't have enough time for grafting. But one thing I wanted to tell you guys before I open it up to more questions, two things. I mentioned resources. These three books are really good resources for looking up information on how to propagate specific species of plants. Hartman and Kester's Plant Propagation is the one that we use at NC State for our plant propagation classes. It has everything about everything that you ever want to know about plant propagation in gory and horrible detail. The reference manual of woody plant propagation here on the far right, that's by Michael Durr. It's set up very similarly to the manual of woody plants, which you're probably all familiar with. So it's listed by species, has really good detailed information about propagating woody plants only. And then finally, this middle book, Creative Propagation. This is a book put out by Timber Press. This has a lot of information about propagating some plants that may not be found in the other books, house plants, herbaceous plants, things of that nature. And it is sort of on it from a more home owner perspective rather than a commercial perspective. So some good resources there. And then finally, a little self-promotion here. If this is a topic you want to know more about, we actually offer home plant propagation. It's a class that I teach. It is available to be taken completely online. So for those of you who aren't local and can't make it to campus, it can be taken completely online, and we are going to be offering that this coming spring semester, spring 2015. So if you want to know more about that, please just let me know, shoot me an email. I'd love to tell you more about it. So with that, does anybody have any questions?
We have time for several questions, so ask away.
All right, I see one here in the chat box. When is the best time to do basal cuttage on bulbs? Generally, it's recommended that you do it either in early spring before they start to actually grow for the year, or to do it right after they've bloomed and they're starting to leaf out for the year. So that depends on if it's a spring flowering or summer flowering bulb. But after it's already flowered and it's leafing out, it's also a good time. You can do it really any time of year, but those two times will have the best success.
Any more questions? We have time for a couple more if anybody has any.
Do master gardeners get a price break? I get that question a lot. Sadly, no. If it was up to me, sure, but it's actually not up to me. So it does cost the same as it does most students, and if you're a North Carolina resident, I believe the price for that is about $650 for the semester. But that is for a 15-week semester, so I feel like you probably get your money's worth.
Okay, questions aren't coming in very fast, so I think I'll thank Liz very much. Really interesting presentation, great pictures, lots of great information. Thanks so much. And if you could hang out for another 5 minutes or so, if any questions come in the chat box, maybe you can handle them as a chat if you have the time.
Sure, that works fine, and thanks for inviting me to talk with you all.
Yeah. Thanks very much for talking. We really enjoyed it. All right, Matt, are you ready?
I am.
All right.
Can you hear me all right?
Yep.
Okay. Well, let's talk about some insects. Okay, so first, let's talk about a robber fly. And robber flies are a really interesting group of flies that are very enjoyable by people. They're very noticeable, and some, like this beast here, are huge. This one is a photo by one of the faculty members in our department, Clyde Sorensen, who took a photo of this large robber fly eating a dragonfly. And you know how big dragonflies are. They're actually, one of my favorite facts is to tell people that there are flies that are known to eat birds and not just suck their blood, but giant robber flies can actually take down hummingbirds, so they're very large. But I'm not going to talk about this type of robber fly today. I'm going to talk about one I saw the other day, right before I mowed my lawn. And these are called, in a nice, affectionate name that I learned a couple weeks ago, gnat ogres. These are in the family Acilidae, as are all robber flies, and they're in the genus Horticulture. Now, we have 3 species in the U.S., in the eastern U.S., widely distributed. 2 of them, Horticulture fusca and Horticulture abdominalis, are very difficult to identify from each other. Horticulture calva being a little grayer than the other 2. But these are very small for robber flies, only about 4 to 9 millimeters long. They're among our smallest, and because of their size and the large eyes that you can actually see here, they're actually one of the more easily identified robber flies. Now, they often perch on grass blades. You sometimes find them around meadows and fields. You'll see them flying very slowly and low around the ground, sitting on the grass blades to perch like many robber flies do. And like other things like birds of prey and such, they'll perch and look out for prey. Now, they are found in open areas and fields, like I said, and usually near swamps or forests. Now, I couldn't find any information about where their larvae live. Like most robber flies, the larvae are probably predators as well, living probably in moist soil or rotting wood, things like that. And as you can see here, this isn't the actual mouth looks like. That's the tail end of another type of fly, a grass fly, that was being eaten by this robber fly. Now, they'll fly around really slow around the grass, and you'll see them sometimes mating. And just to show you a couple more pictures, that's from the front, and of course you see these large eyes. But from the side, they look like a typical robber fly, just smaller. They do have much larger hind legs than the other legs. They've got a kind of large feet and large calves almost on their hind legs. This one right here is doing a service because this is a queen ant. And that ant looks like a Chinese needle ant, which is a stinging ant, now widespread around the southeast. They do sting, and so this robber fly caught this nice queen ant. Will keep her from making a new colony. And so you can tell again how big this robber fly is by how big her prey is. That is, obviously, ants are small, and it takes up a large amount of her mouth, basically. And then if we look at the picture of the other one, I think this is a female, the other one's a male. You can see this is that little grass fly that it caught. And again, grass flies are very, very tiny flies. They're the little eye gnats that you get around your head and everything during the summer when you're sweating or around cuts. So again, these robber flies are very small, and they use their pointy mouth parts to basically pierce the exoskeleton of these small insects. And then they'll go back to their perches, feed on it, suck it dry, continue to hunt for other ones, and then go out and mate and make new little gnat ogres. So keep an eye out on them. They're pretty cute. They're really, they sit there and they posed pretty well. I was able to take several photos of them while they just sat there, especially when they're eating. And just be out there observing them. Notice that they're not biting people, and they actually will catch mosquitoes and other small biting insects, so they are beneficial. Of course, they will eat anything they find, so even some small beneficial insects they may kill. But they're pretty indiscriminate in what they eat, as are most robber flies. Okay, so I just think they're pretty cute, and I feel like presenting on them today. So, gnat ogres. Okay, the next insect I'll be talking about are a group of beetles that I was actually brought these specimens by Dave Steffen. These are yucca flower sap beetles. That's not their approved common name. They really don't even have a common name, but it's good enough because it describes them well. These are in the family Nitidulidae, which are the sap beetles in general. There's some famous members of this group of beetles, including the small hive beetle, picnic beetles, a couple other ones, and I may talk about them over the years, different species, because different species do different things. This species in particular is Carpophilus melanopterus. You can tell it by these, it's a red coloration except for the black elytra. And these are specifically found on the flowers of yucca, Yucca filamentosa. The beetles are attracted to the flowers when they bloom, and the adults and larvae will feed on the flowers. So what happens here is you can see they congregate on the flowers. The adults will nibble on the flowers and mate, lay eggs in the flowers, and the eggs will hatch and the larvae will start feeding. Now the feeding, though, causes the flowers to abscess. So this means they dry up, they fall off, they don't produce fruit. And what happens is they do this on purpose because the larvae prefer to feed on rotting and basically fermenting fruit flowers. So they actually make the environment they want to live in. I've actually read a couple papers on these where there was competition between these and yucca moths because the yucca moths need the seeds of the yucca flowers to develop their larvae, so they're actually in direct competition with these beetles. And basically on a given yucca plant, you'll get some leaves, some flowers that will be fed on by these yucca sap beetles, whereas others will be fed on by the yucca moths. And so the ones with the seeds will develop and be benefited by the yucca moths, whereas the ones that don't develop will be fed on by these sap beetles. Now the larvae will pupate in late June, and the adults will then come out of the pupa but stay in the soil and wait until the following spring to emerge. So there's only one generation per year. And just to show you some more close-ups, so of course the nice mating pair sitting there. Lots of them were mating on this flower, and there were obviously lots in one flower. And they're fairly small. They're about 4 to 5 millimeters long. Now sap beetles in general can be told apart by a lot of other beetles because most species have elytra that are short and expose the tail end of the abdomen. They'll expose at least one to several segments. They also have a clubbed antenna. Their mouth parts are usually exposed and sharp, and they usually also have a very long last tarsal segment. And so if you see these beetles, many of the beetles in this family are attracted to slime flux flows, sap flows, fermenting fruit, compost, things like that. That's why they're called sap beetles. So this species, again, in particular, is Carpophilus melanopterus, is the yucca flower sap beetle. Now, if you do see these on your plants, and you really want the fruit to set, or you want the flowers to stay really nice, you could easily just knock them off the plants into some soapy water. There's probably no need to really spray. Of course, they're only feeding on the temporary parts of the plant, and so you could just leave them be, have them use up some of the flowers, and they're kind of interesting to look at, and they're kind of comical when they're off walking around the plant. Otherwise, they're not going to affect the yuccas, the actual yucca plant's success and health. Okay. Now another thing that I noticed, this was actually out at the landscape field day at the Arboretum. We were walking around, and I noticed this very poor-looking mimosa tree. Very brown leaves, lots of webs everywhere. I collected some specimens, and this is the mimosa webworm. It used to be placed in the family Eponomutidae, but since then the moth phylogeny and moth classification has been kind of shaken up. A lot of these dumping grounds for various species have been split up now into families, and so now it resides, at least for now, in the family Galacticidae. The species is Homodala anisocentra, and these feed only on mimosa and honey locust. Especially as far as honey locust, all the thornless varieties are most susceptible. Now, the insect is actually native to Japan and eastern China, where mimosa is native to, and it was first recorded here in Washington, D.C. in 1943. So it's been around for a while. As the spread of mimosa and honey locust has continued, these moths have also spread. So it's found wherever its hosts are found. So what does it look like? You know, you see all this webbing, you see this unthrifty looking tree. Inside the webbing, then, you're going to have this is a mature larva. You're going to have this greenish yellow with a black collar larva. Now, these are fairly small, they're 1/2 inch to 3/4 of an inch long at full grown, but they are very thin. They also tend to flop around if you try and get them, and they'll flop around and just kind of flop out of their web to avoid being caught by predators. So they erratically kind of roll and squirm. So they're very difficult to collect by hand if you wanted to. Now, the adults, on the other hand, are very kind of pretty grayish silver moth with these black spots all over the wings. This one actually emerged from the ones that I was rearing in the lab. Now, adults fly in late spring. The females are actually attracted to silk to lay eggs. So if you have a tree that was attacked the previous year and you really care about keeping this tree nice and healthy looking, you want to avoid having these moths lay their eggs there, you might want to cut out any of the branches or any of the leaves that have silk on them. Also keeping in mind to clear any of the leaves below the tree on the ground that have silk on them, because this may attract further infestations. Now, the larvae will then skeletonize the leaves and web up the branches. The leaves will turn brown, basically there's 2 generations per year. So the first generation comes out, does a little bit of damage, creates a little bit of webbing. You may not notice it too much. But then late in the summer, late July, early August, you're going to notice the second generation. This is where you get a lot of the more damage, and there's going to be overlapping generations, at least 2 generations per year. Then they overwinter in pupae in silk cocoons on the leaves of the trunk. Now I got a question about, are fall webworms attracted to the silk as well? I don't think so. I'd have to look that up, but I doubt it, especially since locusts and such are not really a very preferred host for fall webworms. So they're going to go after some other more broad-leaved plants and trees. But this will attract the mimosa webworms, obviously. They don't seem to feed on the seedlings, which is a shame because I know I have a ton of seedlings in my yard that I'm always pulling up. And some people don't even really like mimosas, so it might not, But, of course, these are making mimosas look bad, and you don't want a bad-looking tree in the landscape. Otherwise, these moths will do fairly extensive damage to the entire tree, webbing it up and just making it look bad. Now, as far as control, you can try and prune out or find early generations and the silk early in the spring. You can also then, if you have a tree that continues to get attacked year after year, or you suspect it may be attacked, you may try and spray it prophylactically with some BT insecticides or some other types of approved insecticides when you see the moths flying and when you have the very small young larvae of the first generation appearing. Of course, by the time August has come and you've got lots of webbing all over the plants, these caterpillars are hiding in the webbing, feeding inside there. It's very difficult to get contact poisons in there, and I'm not sure of any other types of approved methods for controlling these moths. But I don't know whether this is a widespread thing, whether people have been seeing this in their yards year to year, or if certain trees one year will have heavy infestations, other years will not, and whether this is even a bad year or not. But I have noticed a few trees around that have these, and I've noticed several trees that don't have them. So it's very difficult to predict probably where they're going to hit. Anyway, that's the mimosa webworm. Okay, now for something adorable. I think these are very cute. This is one of my favorite front runners for cutest insects in the world. At least this family, but particularly this species that I found a few months ago while I was outside releasing some other insects. I saw this thing crawling up a tree. Didn't know what it was at first, and brought it inside to look at it closer, and bam, knew what it was. So this is the family Incertidae. This is a type of chalcidoid parasitic wasp. This species in particular is called Chrysoplatycerus splendens, which is a really nice scientific name that literally translates to golden flat horns, and splendens just meaning splendid, very magnificent looking. And you can tell why it's called that. This is a very small wasp, but not as small as many of the chalcidoids get. So this is 3 millimeters long, which is actually kind of gigantic for an inserted, but still it takes a microscope to look at it very closely, or a very good camera. Now these wasps parasitize mealybugs. Inserteds mostly parasitize Homoptera, so scale insects, mealybugs and aphids, things like that. This one in particular parasitize mealybugs, and the known host genera are Dismococcus, Faricea, Formicococcus, Planococcus, and Pseudococcus. So some of the most famous mealybugs are found in those genera. So it's actually a very important little wasp. It has been distributed throughout the world. They found it in Europe. They published a nice paper of the discovery in Italy, and it's basically all around the world where all of these mealybugs have traveled as well. It is likely native to, I think, the tropics and the Western Hemisphere tropics, probably the Caribbean or Central and South America, but we really don't know exactly where it's from, because a lot of these insects that travel around with humans have been traveling around for hundreds of years, basically, and we really don't know exactly where they came from. And since everywhere has them, we'd have to do genetic studies, basically, to figure out where maybe the most diverse genetics are to find where the origin of these insects is. So it has been used for classical biological control for important mealybugs. So people will use it to control these things on commodities and introduce it into other regions of the world. Of course, it's mostly feeding on mealybugs, which we really are not too concerned about going extinct or whatever. I'm not sure how the program is specifically, but I just want to show you another picture of it from the side a little bit more and just describe a little bit of what you're seeing here. So of course the inserteds often have very large eyes, gives a nice cute appearance, I think. Now the antennae, it's very difficult to tell exactly what's going on here to some people, but it's very strange. You basically have this first part of the antenna, which is one segment that's kind of concave and flat. And then the rest of the antenna is segmented, and I'm not sure if it folds in there, but it looks almost like a pocket knife, like it could fold in there. I don't think that's actually what happens. And apparently, from what I've seen, this is only specific for the females. Males will have a typical antenna, flattened, but not as greatly expanded as that one. Now you notice that the coloration is a metallic green to purple blue, and then the scutellum has this golden green metallic color. And then the rest of the pronotum and the side of the body are kind of orange. You'll notice here this looks like a spike, but that's actually a group of setae that are formed in like a spike formation. This is common for some other inserteds too. And it's just really odd looking, I think. Lastly, there's the curved wings. So the wings actually curve down. Now what the function is, I'm not sure. It's probably to keep them streamlined, keep them basically from getting snagged on anything when they're crawling around on trees. And actually inserteds and the closely related family Eupelmidae in the Chalcidoidea, the parasitic wasps, are known for having an enlarged mesothorax that has these nice muscles, and you can see the middle legs are actually bigger than the front and the hind. That's because they use them to jump. And so you'll see these actually walking around on surfaces and hopping around from place to place, maybe flitting their wings just to get a little bit of distance. But really they like to walk around and search for their hosts. They also have this enlarged spur, and that's how you can kind of tell this family, with this large part of the mesothorax, the large leg with the spur, large eyes, crazy antennae. This one also even has little setae on its wings and patches. So looks like a transformer or a robot or something that people came up with, but I guarantee you this is real, and you will find it out there, especially if you've got mealybugs around. So keep your eyes out. Very small, but very cute. Okay. Speaking of mealybugs, we have gotten a few samples lately with striped mealybugs on them. These have been very noticeable when they come in, and pretty large, nice-looking mealybugs. So again, all mealybugs are in the family Pseudococcidae. This species is Ferricia virgata. Now, Ferricia actually was named after a famous scale taxonomist, Ferris is his last name. And basically this is another one where we don't know exactly where it's from, probably in the Neotropics. The genus Ferricia is known mostly from the Western Hemisphere, with a couple species scattered elsewhere, but most of the species are found in the Western Hemisphere. Now again, it's been transported all over the world, and it is also facilitated because it attacks many, many different types of plants. So this species attacks over 50 families of plants. That's just the families. There are many, many genera and species of plants within those families that they attack as well. Now, though they don't transmit diseases here in the U.S., apparently they do transmit some diseases in Africa, especially to cocoa and I think some citrus diseases in Africa. So you don't have to worry about your plants getting sick here, but in other areas of the world they are kind of a serious pest by transmitting diseases as well. Now, striped mealybugs are, like I said, a little bit larger than other mealybugs. They are about 2 to 4 or 5 millimeters long, and basically they the one way that you can identify them more clearly than other mealybugs is that they have the two tails like most mealybugs, but instead of having these basically these fringes along the side, these have little glassy-like tubes or like little hairs coming off of them. And you can see in this picture all these little filaments coming out of the body. So you have these two major waxy tails and then lots of filaments. Whereas most mealybugs won't have those filaments, they'll just have many of these large waxy parts. And that's part of the structure of the insect that causes this, which I'll show you in a second. Now these were some we found on Magnolia. This was the first sample submitted. You can see the young right here that are still developing, the adults right here on this Magnolia fruit. This was Magnolia virginiana. And then we got some grapes with this on there. Now Hannah Barak, our grape entomologist, small fruit entomologist, was asking for people to keep an eye on mealybugs. And she mentioned 3 species. Well, when we got this grape in, I noticed that it looked a lot like the ones from the Magnolia, and so I wanted to confirm. So I cleared it and put it on the slide. Looked under the microscope, and sure enough, it was the same species as was in the Magnolia, and not one that Hannah was had on her radar for grape. So we let her know, and now this is one of the ones that you should be looking out for. Again, it's much larger than most of the other mealybugs. You're going to see, again, lots of little glassy filaments almost coming off the body. Now, this might be a little bit more specific than people want to know, but I'd like to show you a drawing of the cleared specimen because it looks very different than what you would think. So scales and mealybugs are drawn with the dorsal side on the left and the ventral side on the right. And this shows just all the little structures, and this is how we identify these types of insects. So again, it's generally large for mealybugs, about 2 to 4.5 millimeters long. They lack cerariae, except on the anal lobes. Now, cerariae are these little lobes with very stout spines or spikes. Most mealybugs will have them all around the side of the body in varying intervals, and this is what creates the kind of seed or the center for those long waxy filaments, those long on typical mealybugs. Now what creates the long thin glass-like filaments on this mealybug is that they have many tube-like ducts with this oral rim, very hardened and with several seedi off of it. And that's actually how you identify the genus, and particularly around here, the most common species, Virgata, is going to have all these little tubes all over it. And I think that's where these produce these long, waxy, glassy-looking filaments. So again, those are some ways you can tell. Now, I forgot to mention, it's called the striped mealybug because often, when you look at it, it has these 2 lines on the back of non-waxy areas. Now, the specimens I had didn't show that quite that clearly, but there are some pictures and some drawings online showing how the striped mealybugs have very nice, long, unwaxy lines on their back. That may be another way to help identify them, but otherwise you'd have to clear them and such. Okay. Now, I have a little interactive part for people. So Dave Steffen came in the other day, and we were discussing things, and he said he didn't notice many orb weavers this year. And I said, you know, I didn't notice that many either, really, and we're trying to figure out whether this is just something we're noticing or something everybody's noticing. So if you could, I have this broken up to choose your symbol, where you're located, and please mark on the yes or no side whether you've seen more than one orb weaver this year so far. And basically I just want to see whether there's a regional thing or whether it's just not at all what we're seeing. Maybe we just aren't looking hard enough or whatever, but it just seems like a thing. And I'll have one more of these slides after this too. Okay, so we're getting a few no's from the Piedmont, some yeses from the coast. Okay. This is not very scientific, obviously, but I was just curious about what's going on out there. Okay, mountains, haven't seen many. Not sure about the mountains normally during the summer. I would assume there's going to be lots of them there too. Okay. So it looks like, you know, there haven't been that many sightings of them. Now, again, I don't know how much people are actually looking at them, but typically these spiders build their webs right in front of people's homes, right in front of their doors, so they're very noticeable. You end up walking through their webs or seeing their webs every night. But it just seemed like maybe there was something going on this year. Maybe the extra cold winter knocked them out a little bit. But just keep an eye out for them, and we'll see what happens this winter, what happens next year when we look around for these, I think, fairly charismatic little guys, or big guys sometimes, and girls. Okay, so it looks like, yeah, not many people have been seeing them, but some people have been. Okay, again, another one we've been discussing is kudzu bugs, everybody's favorite thing to dislike. You know, again, a little charismatic looking, but really annoying in a lot of ways. So, have you seen more than one of them, or not? Again, this is coming on the heels of people not seeing that many of them this year, as many as other years, and I just wanted to gauge what people were seeing out there. It might be that I'm not getting as many photos of them or submissions of them because everybody knows what they are now. Okay, so people have been seeing them pretty well. Okay. Well, let's see. So you've got a lot of coastal ones seeing it. Okay. Okay, so I see some comments, not as bad as last year, seeing more than one, but less than previous. Okay, somebody's gotten lots of calls for them. So yeah, it may be regional. It also just may be that people aren't seeing that many. Now, yeah, I didn't have the degree as to what people were seeing. Yes, more than one is probably fairly easy to find, but you know, we haven't seen, and some researchers have been trying to find specimens and eggs to actually do experiments on, but have been having some difficulties actually finding them with reliability. Okay, so obviously that means they have not been knocked out completely. They are going to come back, I'm sure, and who knows if it'll be in full force. Who also knows if predators and parasites are starting to catch up with them, but I doubt that that would have such a drastic effect on them this year. I think the cold winter is being the major culprit being blamed for them. Okay. Lastly, a couple of bolos, so be on the lookout. So the first is a request. So last year we got some specimens in of moth and caterpillars that were feeding on Apocynaceae. So they're feeding on Vinca major and Amsonia. And I had identified them as one moth, but upon closer inspection, we figured out that this is actually a different moth that looks very superficially like another moth that feeds on a different family. This is Diaphania, or sometimes called Stemmareis costata. Now, it's often confused with another native species, Palpita kimballii. Now, Palpita kimballii looks very similar to this moth right here, except it feeds on the olive family, the Oleaceae. Now, we had been getting these in from Apocynaceae, and so I thought, well, it's either a new geographical range or it's a new host shift. Well, it was thought to be restricted to the South U.S., basically Texas, Florida, Oklahoma, and it hadn't seemed to be reaching up here. And I thought this was a huge jump in geography. Maybe it switched hosts. Well, what we know now, I've been talking to some researchers in Florida, and we're going to try and publish a paper on the range of this moth, which seems to be maybe more abundant than our actual native Palpita kimballii. So, I'd like to ask if anybody sees a moth like this or caterpillars that are doing major damage on Vinca or Amsonia. Their other host is Confederate jasmine. If you see any of this happening, please send them in because we'd like to have a good record of all the counties in North Carolina, and I'm working with people in Georgia and in Florida to figure out what the range of this invasive species is. Now, there are different reports. The genus isn't even clear. It still needs to be revised, but there are different reports on whether this is actually from South America or Southeast Asia. And it's a very difficult group, and so we have some people in Florida also working on it. So again, here's what the caterpillars look like, the pupae, and the adults. Now last year when I reared these, I also reared out a parasitoid from them. So at least they've got a parasitoid here killing the pupae. And this is a nice calcitid wasp. So anyway, be on the lookout. They'll come to lights, they'll be around, and they'll be around those Apocynaceae. So how large are the larvae and adults? So the adults have a wingspan of about 2 centimeters, about 15 millimeters, so a little less than an inch or so. The larvae are going to be about over half an inch long or so. And this picture is a little misleading. It's light colored here, but they will take on the coloration of their host, so they'll be feeding on green parts of the plant, which will then turn the caterpillar a greenish color. Anyway, and when should we be looking for them? That's just about now. So we got our samples in last year, just about the end of August, early September. So basically this is about the time of year where you're going to see adults flying, where you're going to see the larvae doing the damage. The larvae are going to web up and skeletonize the leaves of Vinca and some of these other hosts. We'd also love to know which hosts you get them from. So if you send them in, please let us know what host it was on, because we'd like to know what the host range is in North America. Great questions. Okay, lastly, a couple last things are be on the lookout for goldenrod insects. Of course, goldenrod is a really, some people think of it as a weed. I love it because it attracts lots of really cool insects. So here you've got a blister beetle. You've got the locust borer, which is a really cool, although it's not on goldenrod here. They are often on goldenrod. There's a longhorn beetle that feeds in dead locusts. This is an elephant mosquito sucking the nectar of one, and here's an ambush bug. And you also get to see soldier beetles, lots of moths and butterflies. Very attractive to lots of insects. Nice little ecosystem where they like to live and sit and suck on the nectar, eat the pollen. Okay, and lastly, just be on the lookout for just the continued activity of many insects. So you're going to have full-grown mantises at this time of year. You're going to have a lot of hornets and other wasps flying around as their colonies reach critical mass. They're going to be flying around and provisioning the colonies and getting ready for winter when the queens will survive. And more. Here's a dogbane leaf beetle, which out in the mountains I saw out on the dogbane. Just lots of insects. It's very difficult to even specify any particular ones because this time of year we're having a lot of activities still. And that's it. Are there any questions? Well, feel free to email me or give me a call if you have any questions. And now I guess we'll turn it over to Mike.
Thank you, Matt. Get something set up here, 1 second. All right, in the, first of all, please give me a green check if you can hear me. See, there's already 3 red X's there, so that, I guess they couldn't hear me. I knew they wouldn't be able to. All right, good. With, thanks, cute. The diseases portion of plants, pests, and pathogens, there's always a challenge, I guess for insects too, as to whether each time to talk about things that people are seeing but that are common, we've talked about before and we're rehashing, versus trying to include some less common things that might be new. And I hope that I achieved something of a balance here today in the presentation. We're going to start out with some things that we have talked about before, and I used the phrase learned from the history instructor as an undergraduate, repetitio est mater studiorum. Repetition is the mother of studies. So maybe a little bit of that here to start with. This was a vinca, annual vinca, which of course should probably be called Madagascar periwinkle in the genus Catharanthus, and it's exhibiting these dead leaves and stems and root rot. Those of you who have been on the program before probably know what this is. Does anybody want to venture a guess in the chat box or put it on the whiteboard? It's probably the most common disease we have on this particular host in our landscapes. Yes, Charlotte, that is correct. This is Phytophthora aerial blight and root rot. I got Phytophthora out of both the roots and the stems of this particular plant. Here's another photograph from our slide collection earlier in the season, not this season, but an earlier stage of the injury. You can see how it attacks the stem, causing a darkened, a little bit sunken area of, we call it necrosis or dead tissue, and then this leads to the leaves also decaying. This is a sample that came in May of this year from Orange County, and you can see how the necrosis or the death of this tissue can be pretty extensive on this plant. Most of the time when you see this in the landscape, you can probably peg it as Phytophthora aerial blight with these particular symptoms. There is one sort of look-alike that we ran into, not this year. This is from Durham County, August of 2012. And immediately it looked like something else might be going on. There's more of almost destruction of the leaves themselves. They're kind of matted together, which we don't see with the Phytophthora. And this turned out to be Rhizoctonia aerial blight. So a very different kind of organism in both cases. Rhizoctonia being a true fungus and Phytophthora being one of our water molds belonging to a group we call the oomycetes, along with Pythium and with the downy mildews. So if there were certain circumstances, particularly in production, where you were going to use chemical control, it would be very different for these two groups. We don't really recommend that, though, for the home landscape. And we would, before we move on to that, let me mention that we do see Phytophthora aerial blight sometimes in petunias as well. For management of I'm sorry, I have another slide here before that even. This was the Phytophthora aerial blight on Erythema, which is one of our aroid plants. This is growing in a nursery here in Wake County that Dr. Brian Whipker and I went out to visit. Interestingly enough, when they saw the concentric ring pattern on the spots, someone at this operation concluded that it was Alternaria solani, which is a fungus that causes early blight on tomato. And it was actually Phytophthora that it turned out to be. In this case, it was quite serious and appeared that the Phytophthora was going down into the bulb, and I actually isolated from the top of the tuber, I should say, of this particular plant or one of its neighbors. So Phytophthora aerial blight, mostly we think about it as a problem on Catharanthus, but it is not exclusive to that. Now, as promised, what do we do about Phytophthora in our home landscapes? The first thing, of course, is to make sure that the plants that you bring in are, in fact, healthy. This is a point that we heard from plant propagation talk at the beginning of the session here, but also when you're talking about transplants, make sure that they are checked for not only stem and leaf problems, but also root problems. Planting in raised beds is a tremendous help against Phytophthora because as a water mold, it requires a lot of moisture. And mulching in this case, since we're dealing with a Phytophthora aerial blight, is helpful because it reduces the amount of splashing of soil getting onto the leaves and therefore infecting, because the fungus, or fungus-like organism, if you want to be picky, is surviving in the soil and it can get onto the foliage by water splash. Of course, reducing your irrigation to what's necessary is another big thing to help against water molds. And finally, try growing something else. The Catharanthus, if the soil is infested, is probably not a good choice there. It's interesting, though, that there are some types, such as the Cora series, which are said to be resistant to Phytophthora. And I believe this is true, but there may be different races of the fungus, so it might not hold up in all cases. Oddly enough, in our own garden at home, we have this plant that reseeds itself every year and comes back, and every year a few plants show symptoms, but mostly it does well, and this may go back to one of the resistant types that we had planted there several years back. The reference there, and I'll cut and paste this into the, one second here, into the text chat box, so you'll have access to it, is to our fact sheet AG 747, which describes in detail the cultural practices that you'll want to use against Phytophthora once it's been identified in your landscape, and also some choices for plants that will have fewer Phytophthora problems. Now, these are not bulletproof. We had, in fact, a case here on campus just this year where they used one of the plants on our list. It was candy tuft, actually, probably not the best choice for our climate here, but it did come down with Phytophthora after it was planted into an infested soil. So these, under high disease pressure, are not going to necessarily give you 100% control, but at least it's a starting point, and the idea would be to avoid those species that are most susceptible. Also keep in mind that there are different species of Phytophthora, and so the plants they affect may vary. Continuing with our theme of repetition, we've got this image sample here on the left from Jones County this month with the symptoms that we would call shot hole on an ornamental cherry. And the picture on the right I took this morning out at Pullen Park. Saw this tree yesterday and went out and snapped the picture this morning. So we are seeing a lot of this. What we weren't aware of a couple of years ago when I first talked about this disease on plants, pests, and pathogens was the fact that it seems that a lot of this is being caused by a fungus called Passiflora circumcissa. So cutting in a circle, and that is what it does. Effectively, the plant is the cutter, though, because it is walling off that infected spot, which then drops out, giving you the appearance that it's either been eaten by insects or blasted by a shotgun, someone getting a jump on the dove season. Now, the problem here, of course, is that you get an aesthetic result of tattered leaves, and the fungus is still probably going to be spoilating on those fallen out spots, so making cleanup even a little bit more difficult. It is an aesthetic problem. It appears that some of the leaf fall that we're seeing on these trees, I would venture to say, is actually a response to the fungal infection as well, but it is not going to threaten the life of the tree in the long run. So we do not recommend any kind of sprays in most cases for this disease, especially since you're talking about trees that can be fairly large and hard to cover with any fungicide. Keeping the leaves cleaned up may help reduce the inoculum, or the amount of fungus carrying over from year to year, but I think, again, it will be interesting to see when the weather turns a little bit drier, will we have less of this disease than we did this year. It seems like the last couple of years we've seen a good amount of this. The reason there are two names listed for the fungus is because, if you remember, those who were on the program a couple of winters ago, we did a little segment on scientific names. And I mentioned then that because many fungi have different forms depending on if they're in their sexual reproductive stage or their asexual reproductive stage. They were given different names, and there is a movement now, and actually a new regulation in the Code of Botanical Nomenclature, that we have to have one name per fungus, and so I don't know which one it's going to be in this case. Some of the details are still being hashed out here, but you may see it as one or the other of these two names, Passiflora circumcissa or Mycosphaerella sericella, going forward. Changing gears a little bit, this is a photograph, or actually two of the 4 photographs that Mac Johnson uploaded from Robeson County this month. The homeowner was concerned about the strange objects in the mulch in the landscape beds. They started out as sort of egg-shaped structures, you can see on the left, and then these orange horns came bursting out of them. The interesting thing here is that the odor was one of the giveaways as to what this was. If you weren't sure when you saw the picture after you read the description, which I quote, "It does seem to have an unpleasant, almost pungent odor," then that really solidifies the identification. Does anybody know what this is? Yes, it's a stinkhorn. Ah, Charlotte, you call an octopus stinkhorn. That I've never heard that particular name before, but one of the possibilities is that it was this or something very similar to it. I took this picture back in January of 2008 here on campus, where the multiple horns coming out of that individual egg, we call it actually an egg stage, are fused at the top. And I'm not sure if in the Robeson County case there was, just because it was older, it had broken up. But this particular one here, when I saw it and smelled it, the odor was pretty strong. I would say that it smelled like dead fish wrapped in a wet diaper. There are other different species of stinkhorns that we will occasionally see. Here's a picture I took this morning in a mulch bed around a tree along Dan Allen Drive here on campus. This one is Phallus ravenellii, a different kind of stinkhorn. And you can see at the very top this area called the gleba, which is where the spores are produced. And the odor, foul to us, is actually pleasant, or attractive anyway, to flies. And Matt can talk about what kind of flies like to jump on these, but those are the method that the fungus uses to spread its spores. So the flies are attracted to the sticky mass, and as they leave, they carry the spores along with them. Of course, the question that folks have is, what can be done about these fungi coming up in their mulch? And unfortunately, there's not a lot that can be done. The odor problem, of course, you can take care of by removing them as soon as they come up, but there's not a whole lot you can do. You could try turning under the old mulch and replacing it with composted mulch or coarse pine bark, and that may help. And I'm referring here to not just the stinkhorns, but just the nuisance fungi in general. Remember, we've talked before about artillery fungus. We may have talked about bird's nest fungi in the program. And, of course, the stinkhorns would be another one, what we would consider nuisance fungi, because they're not really harming any plants at all. They're just unpleasant to the people who are trying to enjoy the landscape. Interestingly enough, actually this is kind of counterintuitive. We always insist, if you want to have fewer problems with fungi, use less water, make sure things don't get wet, etc. But it turns out that at certain critical stages of the composting process with mulch, making sure that there's enough water is actually helpful. What happens here is that the moisture allows bacteria to colonize the mulch ahead of the invasion by the nuisance fungi, and these bacteria will suppress the colonization by the fungi. There are some interesting other details on this, and I will is a link on one of our blogs that we did back in November of 2012. I'll just copy the reference to the blog itself. On nuisance fungi and mulches, and there's a link to one of the folks at Ohio State who talks about the microbiology of the mulch and how this affects whether we get different problems going forward. There's also a link there to some work at Penn State on the artillery fungus and what to do about it. As our next and interactive portion, I wanted to do something a little different. I grabbed some things out of the garden at home to try and bring and put on the program, but it turned out that all three were different from what I expected. When I picked them, I thought one thing or two, got them into the lab, and they turned out to be something else. So I set this up as sort of a quiz, and what we'll do is I'm going to change the polling to A, B, C, although it's only going to be A and B in each case. And if you look under your name, what had been a little check mark should now look like a small case, a letter A in a box. And that's where, if you click on that or you hover over it, you'll see you'll have your A, B, and C choices. And in this case, I see someone's already selected A and then deselected it. So that's what we want to do, but it's only going to be A or B. So what I've done in each case is put a picture of the problem and two choices, one of which was what I thought the problem was, and the other of which was what it turned out to be. Well, by the way, that is not a typo. My wife in her nickname just uses one T. So let's begin. What is causing the spots on this zinnia? Is it the fungus Alternaria, or is it the bacterium Xanthomonas? As you're making your choices there, let me add that I haven't 100% confirmed it as the cause of the spotting on the petals, but on the leaves there's really no question. Can't say much more or I will give it away. Give folks another second or two here. All right, let's see. According to you folks, the 7 out of 50 said it was Alternaria, 16 out of 50 thought it was Xanthomonas bacterium, and 27 out of 50 did not reply. If that was because you were unsure, that's fine. If you couldn't find the polling, then let us know afterwards so we can help you find the features of the Illuminate interface. Well, it turns out that I thought it was Xanthomonas as well, a bacterium, but the actual answer here was it turned out to be Alternaria. On the right, you'll see the spores, the microscopic spores of the fungus. You would not see these with the unaided eye. I just include them for interest there. We use them in diagnosing them in the lab. The size of these, the width is approximately the same as the width of a human hair, so they are quite small. They're very dark though, so if you have a hand lens and there are a bunch of them on a spot, you can actually see them a little bit. But for practical purposes, what should have been my clue that I was looking at Alternaria instead of Xanthomonas? Because those are really the two big choices that you would have when you see this particular symptom on zinnia. And what I should have done was held the leaf up to the light and let the transmitted light show me that those spots look fairly dry. There's not a water-soaked margin, kind of a translucent margin around the spots, or within them, that we would expect to see if this were a bacterial disease. That's often a helpful clue if you're trying to diagnose bacteria. The fact that the spots were a little bit angular, you can see here this one stopped with a vein, that's typical of bacterial diseases, but not exclusive. So that, I'll just say that that might have been one of the things that threw me off. I should mention here, while we're talking about Alternaria, is that not all, all Alternaria species are created unequal. I don't think that came out right. What I mean to say is that there are many species of Alternaria. This one is Alternaria zinniae, and it will only affect zinnias, so it's not a threat to your tomato plants, for example. Likewise, there are Alternaria species that are pretty much saprophytes. They're living on tissue that's already dead and not causing any primary damage. So there's going to be a lot of variation when we use the term Alternaria. Be aware that you should drill down for specifics and ask questions. Next question. What is causing spots on bean leaves? I didn't include bacteria here, although you might suspect it. In fact, that was one of my hypotheses when I picked it out. But I'm only offering 2 fungi for your consideration. Is this Cercospora or is this Calototricum? Let me clear off, hold on. Clear the old responses off here. All right, and start again, please. Cercospora versus Calototricum on bean. Folks are a little more timid on this one. Let's give you about 5 more seconds there. Oh, no, I'll give you more because I can hear there's groups of people fighting out in the counties. Disagreements as to which to post. Okay, let's see. So in this case, the majority of those who replied, a small majority, went with Cercospora and a minority with Calototricum. Well, I thought it was Calototricum too, and it turned out it was Cercospora once I got it into the lab and looked under the microscope and saw these spores. This, again, is highly magnified. You could fit, depending on which way you put them in there, you could fit 2 or 3 of these things inside of a hair shaft lengthwise. And this shows us that it was, in fact, Cercospora. The name of the disease, according to the 2nd edition Compendium of Bean Diseases, is Cercospora leaf spot and blotch. It's a pretty appropriate name here. And it's interesting that it comments that little is known regarding the disease cycle of these pathogens. The fungus contaminates seeds and apparently survives in crop debris, so you can believe that these plants will not be going into the compost pile. Vigorously growing leaves rarely become infected, so I'm hoping that this is a case of late season, plants kind of petering out, and this jumping in. I observed similar symptoms last year, so it's not something that I'm going to try and treat. There are apparently differences in susceptibility among cultivars, but I don't have a list of which ones would be more and which ones would be less susceptible. If someone were to become extremely desperate, the fungicide chlorothalonil should be effective, but I would not actually recommend that as being unnecessary. And since you're going to be wanting to harvest pretty frequently out there, that's something else to be considered. One more. Why are these tomato leaves looking blotchy? I'll offer you 2 fungi again. Stemphilium or Septoria. Oh, I'm sorry, I keep forgetting to clear the poll. So hold off 1 second. Let's see. All right, now go ahead and vote for A or B. And it's neck and neck. Septoria was running ahead, but Stemphillium made a surge at the end and crossing the finish line. Well, don't feel bad again because I thought it was Septoria too, but I couldn't find any Septoria on these leaves when I got into the lab. What I did find was Stemphillium. In particular, this was Stemphillium solani. The disease is called gray leaf spot, not to be confused with gray leaf spot as a common name for the disease on, for example, crabgrass. It's not the same fungus. There are, I was looking up information on this or trying, there are some commercial cultivars available that have resistance to this disease. You can look those up in the Ag Chem manual, but I did not go and try and find if there are listed resistances in cultivars available to home gardeners. I don't know either whether the common home garden sprays would be effective here. This is Roma tomato, as you can see, and it seems that there are fewer cultivars of Romas available than the round-fruited tomatoes that have resistance here. So let's be on the lookout for a couple of things. We've talked about box blight in the past. Here's a picture that Dr. Kelly Ivers took in the mountains. You can see that it is a disease that causes widespread defoliation of plants. And let me just zoom out on this photograph and show you the context in which she took this. It was the destruction of these plants at a nursery in the mountains in December of 2011 to try and stop the spread of what at the time was a new disease in North America. It's not just in North Carolina, it's in quite a few states and some Canadian provinces. Up until recently, meaning up until this summer, there was only one find that we had in North Carolina outside of the mountains, however. And then this summer something happened. Someone was selling plants at the farmer's market from his nursery in the mountains and ended up actually selling some infected boxwood plants that had box blight. So we're asking folks to be again on the lookout for this particular disease. You may see the defoliation, which tends to go from the bottom of the plant upward, but in the earlier stages of the disease, look for these sort of chocolate brown spots, which may or may not start at the leaf tip, and for the very characteristic, although not always associated with box blight, dark streaks along the green stems. So if you do see this particular disease, do bring it to the attention of your county cooperative extension office. By the way, those spots do show up on both sides of the leaf blade. Let me go back to that picture again. You can see it's on the upper surface, but also visible on the lower surface. There are some things that you can rule out as being box blight just on the fact that they only are visible on one surface. Other things to be on the lookout for are the general phytophthora diseases, which we could still be seeing in the landscape. Powdery mildews are still out there. The same zinnias that had the or zinnia neighbors of them had the alternaria had powdery mildew as well. Root knot nematodes, we got a sample in yesterday, no, it was last week, that had extensive root knot nematode injury. They had applied in its commercial landscape fungicides every month, and they had 50% or 40% problems, and it turned out to be root knot nematode. And as a general thing to be on the lookout for, school children have returned to classes as of yesterday around here and in many places in North Carolina, so keep in mind that they will be on the roads more than they had been during the summer. In woody ornamentals, this is the time of year coming up where if armillaria is present, it may, but does not always, produce the cluster of honey-colored mushrooms, indicating its presence. Trees and shrubs can decline from this disease at any time of year, but only for a short window in the fall will the fruiting bodies be visible, and they will not always be visible. We'll also be seeing the fruiting bodies of the wood decay fungus Pseudoinnonotus dryadius, especially at the bases of many of our oak trees. This indicates that there's a decay going on in the wood of that root or bud. Passiflora needle blight on spruce in the mountains, something to be watching for, and Rhizosphaera needlecast in spruce in the mountains. Passiflora needle blight on leyland cypress and other Cupressaceae, which we may be seeing in different parts of the state. And of course, continued problems with poor planting techniques may be showing up. In our flower beds, be aware that we're getting into pansy and viola season, and one of our main problems there in terms of diseases is black root rot. Please be sure and check those transplants if they have healthy root systems, and also avoid the temptation or resist the temptation to plant them before the weather has cooled off enough for these plants. And late season leaf spots, as we saw also in our talk just a few seconds ago. With that, I will be happy to take any questions. Actually, let me post one more link for you where we've got our information on box blight. It's the, sorry, here we go. It's the link to our ornamentals plant pathology extension page, but on there you'll find several publications referring to box blight, its identification, susceptibility of different types, and so forth.
All right. Thanks, Mike and Matt. Any questions for either of them before we sign off? All right. You can always reach them through the clinic if you have questions about this or something else. So I want to thank all the speakers today. Very interesting presentations. And we'll be seeing you for the next session on October 28th.
All right, in BJ's words, I just kicked everyone else out.
Thanks, Mike. Bye.
Anytime. Oh, someone else just joined, though. To make sure. All right, Margaret. I don't know if you can hear me, but we have to... Oh, okay. I guess we're good now. Signing off.